1 second. I just got a friend to bring out here. So, what sex is this fish? Is it male or female? We can look inside and we can figure that out pretty easily. But if we look deep inside into its genetic code and ask that question, well, good luck. That is a mystery that's still unraveling. We think of salmon sex like we do for humans. There's X and Y chromosomes, and males have a Y chromosome. But salmon chromosomes aren't well developed. You need something more, a mechanism. Introducing the SDY protein. It's coded for by the SDY gene, about 4,000 base pairs, and thought of to be the master sex gene in salmonids, recently discovered in 2012. This gene is essentially a switch for males. It creates the maleness. It turns on in the developing gonads of a fish, if it's present, causing a cascading kaleidoscope of genes and hormones, ultimately forming testes. Yay! But I didn't know anything about that. You see, I didn't care about the SCY gene. I just wanted to sex this fish, the noble steelhead, to see if males or females had different resistance to a virus, IHNV. You can see the nasty effects below. I thought my research would progress like this. We would take the samples, sex the fish, do some mathy stuff, as scientists do, and I would find differences and get published. Yeah, that's never how things go in science, okay? I was in for a shocker. To sex the fish, we targeted a section of the SDY gene. We did some science stuff. We duplicated the gene, or the section of the DNA thousands of times, billions of times, and then we attached a dye to the DNA so that we could actually see it in a gel. This is what I was looking for phase 2. Females, 1 band, males, 2 bands. That extra band that you're seeing is literally the SDY gene shining back at us. So, that's not what I got. This is what I got. Quite messy. We're looking at 2 bands almost every time, all males. All males is bad. My test was not working out so well. So I thought of the possibilities. The samples could be contaminated, likely my fault, being the dumb undergraduate in the lab. They're all males, in which case my experiment could be over, or the test had failed. The most intriguing and terrifying possibility. We thought we had done something wrong. So we considered all these possibilities. We tried different cycles, different temperatures, different gels. If you're wondering what that blue gel is above, not a clue. It's a messy process. Finally, after exhausting all the options, we came to this. I extracted new DNA from known male and female adult Onchrycus micus and got the exact same results. 2 bands for the males and the females. The SDY gene was not going away. So I decided to make a new test. So I spent almost my entire capstone undergraduate project working on this new test, targeting different sections of the gene, using primers, trying to get a nice result. Finally, after months of work and weeks away from my graduation, without a paper written, I got this. 2 beautiful bright bands and 1 band for the female. But you're all celebrating, but it's not so good. You see, it's not conclusive. If you look closely, the females, almost all of them, have a tiny speck, like a UFO sighting or something. This smudge of genetic information was saying something. What does it mean? I believe it means that the females contain a homologous SGY gene, meaning very similar. Our test was finding it buried within the female genetic code, something that shouldn't have been there. The male sex gene. What is it doing there in the females? No clue. We don't know. But if you think of the salmon sexual strategy, it's a lot like our strategy, actually. Gonachronism, what we're familiar with. But it's just one out of a plethora of possibilities. And even that, seemingly so simple, we don't understand. There's more to it. And males and females, not as different as we had expected. We had clear expectations. 2 bands, 1 band. Males, females. What we got instead was imperfect, but wildly more dynamic and diverse than we had thought possible. We have a lot to learn about how the SDY gene works, but I did learn that salmon sex identity is really diverse and doesn't follow classification. It's not so black and white like I wanted it to be. And that's something as humans we can all appreciate too. Thank you. I am Trevor Eakes. I will be the smelly fish guy. My mentors are Dr. Carrie Nation Marine Brook, funded by the University of Washington. If you have questions, go to the bar or email me, mysid@uw.edu.
Yes? No. I want to borrow the first one.